Hey guys, um, sorry, we're going to be doing white blood cells today. So let me just go right into that. Get back up here to the top. All right, so I'm filling these out. Neutrophils. Oh, hang on, let me get my pen working. Okay, neutrophils, this should be D. These are the most abundant white blood cells and usually the first responders to bacterial infections. Then we have eosinophils, and these are A. They're what you're going to see more of if you're fighting a parasitic infection. Basophils are E. They're the rarest type, and they're associated with the histamine response, or more of the response that you get when there's an allergic reaction, your body's having allergies. Lymphocytes, this is C, right? Yep, responsible for specific immunity. Lymphocytes mature into either B cells or T cells. And these guys are important because they're kind of the assassins of the white blood cell group. In other words, they have specific targets that they go after. Monocytes, like these guys, they're the type that are going to turn into macrophages. Largest of the white blood cells, meaning in size, these guys are big. Monocytes will, or macrophages, will kind of go after anything that doesn't belong. Whereas again, the lymphocytes, as they mature into B and T cells, have specific targets that they're going to go after. Okay, we'll get into that a little bit more in a minute. Okay, so guys, I'm going to do something a little bit different. As I probably mentioned, as you probably already know, there's a bunch of us that teach this 1200 class, and we all have the same notes. And so we're all really careful to make sure that we provide you with the same information on your notes, that everybody else is getting. What I'm going to do is actually fill out those notes, and then I'm going to go over them a little bit just by talking about it to explain it, okay? It's a little bit more freeing for me to be able to do it that way versus constantly trying to make sure that I'm talking in order. So having said that, here go these notes. White blood cells are also called leukocytes. Just like red blood cells are called erythrocytes. These are leukocytes. They function in disease prevention, part of the immune system. Did I fit it? Not really, but we made it happen. They arise from stem cells in the red bone marrow, just like red blood cells do. And then red blood cells have something called EPO that stimulates their production. And white blood cells have something called colony stimulating factor. And this also helps in the differentiation process when it comes to the cells taking on their special structure and function. Okay, I'm going to stop for a second. So what I want you to look at, guys, is this little diagram here. You can see the stem cells, which we would say are in the red bone marrow. And notice that they give rise, these stem cells do, to 4 different types of cells, kind of these intermediate stage cells. Way over on the left of the screen, you've got your erythroblasts, which are going to become the erythrocytes, or the red blood cells. On the far right, you have something called megakaryoblast, and these are going to become the platelets, okay, platelet component of blood. Then you have 3 different types that are going to become white blood cells. They're called lymphoblasts, monoblasts, and myeloblasts, okay? Lymphoblasts become lymphocytes. Helpful, they share the same prefix. Monoblasts become monocytes, also helpful, okay? Myeloblasts, however, are going to break into neutrophils, eosinophils, and basophils. And neutrophils, I don't think were on that worksheet. Well, of course they were. I don't know why I didn't think that. Anyway, so those are going to differentiate into these different types. There's a little bit more information underneath what they do, some of their distinguishing characteristics. You will need to know what each type of these white blood cells targets against when it comes to exams and quizzes and stuff like that. But like the eosinophils, you shouldn't get any questions asking if it contains granules or not, okay? All right, so let's move on to how white blood cells fight infection. The first is through a process called phagocytosis, which can be equated to cell eating, quote unquote. Really what happens is the white blood cell will engulf foreign matter. And then lysosomes break down the foreign matter. All right, so cells don't have mouths, right? Hence the eating in quotations. What they do is there's this invagination that develops in the cell membrane, which if you remember from when we went over muscles and nerves, invagination is like this indentation in the membrane. And so the membrane then, the outer membrane of the white blood cell will actually come out and wrap around the foreign substance. The two ends of the membrane then will fuse back together and you have something called a vesicle. And a vesicle is a little membrane enclosed pouch, okay? In this case, the vesicle contains foreign matter. That vesicle moves to the lysosome, which if you remember from our organelles, the lysosome is the organelle that has digestive enzymes in it, okay? So the vesicle of foreign material moves through the white blood cell, merges with the membrane of the lysosome, and empties out all of its contents into the lysosome. The lysosome is then going to break it down. Need to get a drink. Okay? All right. The other thing that they can do is produce antibodies. Let me write these notes down. Okay. So antibodies are specialized proteins that are attached to the surface of cells. We'll talk about them in the context of white blood cells. They're on the surface of white blood cells, and their job is to bind to foreign substances, okay? This is a chemical process. It's easy for us to think about these things, you know, physically, so we can think about them reaching out and grabbing a foreign particle that doesn't belong. Really, it's a chemical reaction that takes place between the binding site of the antibody and the binding site of the antigen, okay? They hook together chemically. When they are there, it triggers phagocytosis, which we know then the big guys are going to come in and consume it and destroy it. When I talk about the immune system, which we're going to be doing this week, so I will say it now too, I always think about the immune system as kind of like the different branches of military, okay? And I think about fighting disease like it's a war. A lot of those metaphors, if you will, translate well. So when it comes to the antigen thing, this is a bit of a gruesome example, but what happens is when a foreign substance is taken into the white blood cells and broken down by the lysosomes, the specific antigens, the part that are going to cause disease, they get sent back up to the cell membrane where they're going to be displayed on these antibodies. Essentially, this is the equivalent of what do I want to say? This is the equivalent of an army putting all of its enemies' heads on poles. Okay, boom, there they are. These are the bad guys. Then those cells that are carrying those antigens circulate through the body. And as they do, they alert everybody else, all the other white blood cell soldiers, to the fact that these things right here, this head on a pole, is the enemy. Look out for this. When you see more of them, destroy it. Okay, so that's how this works. In a simplified nutshell. Let us move on. Leukocyte disorders, there's something called severe combined immunodeficiency, or SCID. This is the boy in the bubble disease in the 80s. And in the early 90s, I'm gonna look on the dates on that specifically. But basically, that's kind of what there was. A boy had this disease and would catch anything and everything that came along. And so in order to keep him in a sterile environment, he essentially lived in a bubble. What happens with this disorder is that those lymphocytes that we talked about that become B and T cells, well, they don't mature, okay? So they're missing an enzyme that turns those from those kind of precursor cells into assassins, right? So that doesn't happen, which means there's nothing there to recognize specific invaders. All right, what does that mean? Well, most of you have probably been vaccinated at some point in your life for something. Okay, vaccines are made by taking viral particles and injecting them into our bodies, right? Those viral particles alert the immune system to a possible future invasion, okay? So it says, hey, here's all the stuff that could invade you. I'm giving you a warning. I'm telling you what they could look like. You haven't seen them yet, but they're probably gonna come, and this is what they look like. It gives the immune system or the troops a chance to assemble and prepare prior to an attack. Then, if you encounter that virus in real life, the immune system is already ready to fight it, okay? Because normally the immune response can take 7 to 10 days for your body to ramp up enough to kill it. That's where those B and T cells come in. Because if you've had a vaccine, the B and T cells are already looking for it as the assassins. And SCID, they're not there, they don't mature, and therefore there aren't any assassins on the loose. So treatments for this now include regular injections to provide the body with what it needs, and bone marrow transplantation. Then we have leukemia, which is cancer of the white blood cells. Causes an abundance of immature, abnormal, mutated white blood cells that cause all kinds of problems. Bone marrow transplantation can be a treatment option. Sometimes you hear. All right, let's talk about blood typing and transfusion. Again, let me make sure I get these written down first, and we'll talk about it. Isn't it exciting to just sit here and watch me write? Okay. On the surface of our red blood cells are these protein markers, specifically the glycoproteins. And there are two specific types. Okay, you have your type A and your type B. And makes them different is chemical structure, composition. Okay. You have 4 different options when it comes to blood type. You can be type A, you can be type B, or you can be type AB. Those are the ones that have glycoprotein markers on the surface. Type AB blood is an example of codominance in humans because if you pair A with O, if you pair B with O, the A is going to be positive or dominant. The B is going to be dominant. But when you put A and B together, they share dominance, codominance. The only type of red blood cell that does not have any glycoproteins on its surface is type O blood, okay? Type O has nothing. So there you go. I want to point out that these glycoproteins are often referred to as antigens. Now this can seem confusing because we just said that antigens are any foreign substance that can trigger an immune response. Well, if you get the wrong blood type in your body, into your body, that blood is an antigen. More specifically, the glycoproteins on that blood are antigens. So sometimes you'll hear it said that the red blood cells have antigens on the surface, okay? It's kind of like it would be an antigen if you put your blood into somebody else, okay? Just so you're not confused. Oh, well, maybe that made it more confusing. Just you often hear it talked about the A antigen on a red blood cell. It's these glycoproteins. All right, let's talk about compatibility. Again, let me make sure we get these notes down. We already talked about what antigens are. Wrong type is seen as an antigen. Your body says that doesn't belong here. Okay, so the immune system recognizes the cells that quote unquote belong to it or that belong to the body, okay? So if the immune system, the white blood cells are circulating in the body and they see something that doesn't have a marker on it that says belonging to Kelsey or belonging to whoever, then it gets tagged for destruction, right? Calls in the big guns to come and destroy it because it's foreign. These self markers are kind of like having a name tag on all of your stuff, okay? And so if you get a blood transplant from somebody else that has the wrong antigen on the surface, then the body says, that doesn't belong to me, does not have my name tag on it, has somebody else's name tag on it, we are going to destroy it. Body's a little possessive when it comes to keeping what belongs to it. Okay, so here's that word antigen again in the use of the glycoprotein markers that are on the surface. Type A blood is going to have a type A antigen, which means it has an antibody, which are the things that destroy antigens, to type B, okay? Then we have type B blood has a type B antigen, which means it has an antibody of A. AB blood has antigens of both A and B, which means it doesn't have any antibodies because they can get A or B and it's like, it's all good, it's part of me. Then again, we've got our O blood that has nothing on its surface, no antigens, and it has antibodies to everybody. Bummer, okay? O can only get from O, but O can give to everybody. O is a giver. Okay, so let's come down here to our donate and receive. Type A can donate to either A or AB, and it can receive from either A or O. B can give to B or AB, but it can only get from B or O. And again, since O doesn't have any of those surface antigens, there's nothing there to say it doesn't belong. The recipient's body doesn't mark it as being foreign. Okay, AB can donate to type AB, but it can get from A, B, and O. This makes them a universal receiver. They can get from any blood type. A, B, and O, there you go. O can donate to everybody, A, B, and itself. But, bummer, it can only get from O. So who's the universal donor? O, okay? Makes sense? Hopefully you're saying, yes, yes it does. If it does not make sense, email me, let's talk, for real. Okay, obviously we're not going to be doing this group activity, but let's go ahead and talk about the RH factor or the rhesus factor. RH factor is either there or it's not. And it's another marker on red blood cells. So not only would your red blood cells have the markers that say whether it's type A, B, or AB, but it would also have RH factor markers or not. Okay? So if RH factors are present on the red blood cells, you're said to be RH positive. If there are no markers for RH factor on the red blood cells, then you are RH negative. Okay, so either exists or it doesn't. This really only comes to be a problem with RH negative, okay? All right. So as long as a person is given blood from the same blood type, but also the same RH group, then there aren't going to be issues. But if you give somebody that's a negative, a positive blood, that's going to cause a problem, because the presence of those RH factors on the A positive blood is going to trigger an immune response in the person who's A negative. Okay, that's a bad thing, because you still end up with the same clumping immune response, which shuts everything down and leads to death. Okay, so an RH positive person can donate to other people that are RH positive, but they can receive from either. Okay, RH negative person can donate to either, but can only get from negative. One of the most common issues, if you will, with RH negative or with being RH negative, really it's if you're a woman, because it can complicate pregnancies, especially second and third pregnancies, or you're at the lowest risk for complications usually with a first pregnancy, but subsequent pregnancies, the risk increases. Because if the mother is RH negative, but the fetus is RH positive, then what happens is at birth, when blood from mom and baby kind of commingle, the mom can be exposed to that RH positive factor. And her body is then going to begin to create antibodies against the RH positive factor. Okay. Therefore, if the next fetus is RH positive also, it will be exposed to the RH antibodies that were made from the first birth. And this can lead to death of the fetus, and frankly, sometimes of mom too. So what do they do about it? Well, ladies, it's fun. There's something called RhoGAM, and it's an injection that's given to RH negative mothers. It says within 72 hours of giving birth. And what it does is it removes any of the RH factors that may be there so that the mom's body does not make antibodies. So that then there's not that risk in subsequent pregnancies. My sister is RH negative, and throughout her entire pregnancy, she was given RhoGAM shots. I know that they can do it within 72 hours after, but a lot of times they do it once or twice during the pregnancy as well, just in case. Okay. Actually, it was the death of moms and babies during childbirth that led to the discovery of the RH factor, because doctors were trying to understand why, especially for a mom, if she was given a blood transfusion, why she had A blood and she was given A blood, why was the blood clumping just like they'd see if they gave the wrong type of blood? And then how come they were seeing these types of issues in fetal death with this exchange of blood? And so led to more research, and they actually discovered the RH factor. And I want to say that was actually only in like the 1930s. So it's been within a century, which may feel like forever long, but it's kind of not. Why is the immune system so ineffective against cancer cells? Because for the most part, cancer cells are still marked as belonging to the body. So the immune system passes over them because it's like, hey, it's got my name tag on it. It's kind of funky looking, but it's got my name tag, so we're good. There are some immunotherapies that are trying to take cells that do recognize cancer and send them into the body to go and hunt those cancer cells down and destroy them. Okay, awesome. If you have questions over any of this, please let me know. As always, sooner rather than later, just because the end of the semester is rapidly approaching, guys. We've got Thanksgiving here in like, what, 2.5 weeks? So don't put it off. All right, I will see you soon.